chapter 11. We're going to spend most of the time on chapter 12. Chapter 11, I'm just going to kind of touch on some things, so let's do that. Okay. So here is chapter 11 on the muscular system. If you have a question, since I have it in slideshow mode, I won't be able to see the chat. So I'll, between the two chapters, I'll check for questions. If you don't want to wait for the end of the chapter for an answer, just unmute your mic and ask. That's no problem if you have a question. So let's go with the muscular system. I'm going to just skip to this slide right here, where you should know the functional groups of muscles and function according to what they're doing. So what we mean by a functional group, a muscle could be a prime mover. That means when there's a specific movement on a joint, there's more than one muscle that's producing that movement. But the muscle that has most of the load or the major responsibility is called the prime mover. And the muscles that we go through are mainly prime movers in the body for a specific movement. The antagonist does the opposite. So when a prime mover is performing its movement, the antagonist opposes it. So if one muscle is contracting, the antagonist is being stretched. Now we can reverse that, and that antagonist can become the prime mover, which would make the original prime mover the antagonist. So one muscle doesn't necessarily just fit in one of these categories. One muscle can be any of these, or all of these, just at different times, depending on which movement is being performed. So if a prime mover is performing a movement, well, there's synergists that are helping it. So they add some extra force to the same movement. And then we have some muscles that stabilize a joint or immobilize a bone, like it says, while you're using the prime mover, and those are called fixators. So we can see, I don't have a picture there. So the main thing here is the prime mover and the antagonist oppose each other. If you're asked for an antagonist of a muscle, well, then we should know the action of that muscle, so the antagonist will perform the opposite action. So if a muscle flexes the elbow, well, the antagonist will extend the elbow. Right? If a muscle abducts the shoulder, the antagonist will adduct the shoulder. So, all right. I think we're good there. I'm going to skip through naming, arrangement of fascicles, and kind of pause here. I know I skipped through a bunch. That's just reading. I'm not going to read it to you, but I want to go through the muscles in the body. Of course, there's over 600, but we don't come anywhere near that. We don't have to worry about origin and insertion. When we're going through muscles, we're going to be most concerned with their groups. So what muscles belong to certain groups, which I'll kind of bring up as we go, and certain muscles will be concerned with the actions. We label muscles in lab. We spend a lot of time in lab labeling all these same muscles on the models. Of course, on the lecture test, I don't do any labeling, just like with bone. We just stick to other concepts because you have enough labeling and pictures and that kind of stuff for lab. So let's kind of start going through it. Some of these you recognize, well, all of them you should recognize from lab, like the epicranius or occipital frontalis. That moves your scalp. Fine, there's not a whole lot to say there. These are muscles of facial expression. So these you should recognize as being the muscles of facial expression. The smile muscles, which are the zygomaticus major and minor. The grimace or the frown muscles, which is the depressor anguli oris. The muscles that close the lips, orbicularis oris, and closes the eye, orbicularis oculi, and works the cheek or compresses the cheek, moves the cheek, buccinator. So I'm assuming you know all of these already because we spend a lot of time in lab on them. So I'm going to move to the next group. And these muscles on this slide right here are your muscles of mastication, which is chewing. So we have the prime movers of closing your jaw, the masseter and temporalis. The pterygoids, median lateral, perform a grinding movement side to side. And then our buccinator, also our cheek muscle buccinator, also helps maneuver food in our mouth. So it has a chewing role as well. So all of these muscles, all 5 of them, could be considered muscles of mastication. So you should be able to group these as muscles of mastication. And I'm not going to really show many pictures here because that's lab material. The digastric and mylohyoid, sort of under the chin there, swallowing in speech, and that's fine there. The sternocleidomastoid and the 3 scalenes, anterior, middle, and posterior, those are the muscles that we learned that move the head, work on the neck to move the head in different directions, and if pretty much covers almost all of it except extension. See, major head flexor, laterally rotates, flex. So moves that, except for head extension, performs all these 2 muscles together, form all other head movements. The erector spinae group extends the back, right? Those are postural muscles to hold you or to keep you straight or upright. The muscles of respiration, there's the 3. So number 1, know these 3: diaphragm, external intercostals, and internal intercostals as muscles of respiration. But then go one step further here. Respiration is 2 parts, right? There's inspiration, breathing in, and expiration, breathing out. Well, when you're breathing in, you're contracting your diaphragm and your external intercostals. So the diaphragm and external intercostals are for inspiration, breathing in like you see there, right, and there. Breathing out is just relaxing those muscles. So we don't, expiration is not an active process, it's a passive process. So these 2 muscles, diaphragm and intercostals, that just contracted for inspiration, then relax for expiration. However, we could, if we wanted to, force air out of your lungs, like blowing a balloon, even when you speak or sing or whatever reason you're forcing air out of your lungs, you can do that. So if it's not regular expiration, if it's forced expiration, then it's the internal intercostals. So a little tricky because the external is for inspiration and the internal is for expiration. So like the ins and the x's kind of alternate. But remember that expiration is forced expiration, not regular expiration. And I'm not worried too much about pictures here. These are the 4 muscles that make up the abdominal wall, which we learned in lab. So recognize these as abdominal muscles, where 2 of them have the term abdominis in it, which makes it pretty simple. And they exist in 3 or 4 layers. I'll go to the picture here, right? So the external oblique is the outer layer. I should say 4 muscles, but in 3 layers. External oblique is the outer layer. The rectus abdominis and internal oblique make that middle layer. And then the transverse abdominis is the deepest of the 3 layers. Pectoralis minor, I would just worry about labeling that in lab for the final. You already had your quiz on it if you're in my lab, but it's just such a complicated action that I probably would not give you a pectoralis minor action. The serratus, by the way, you might have realized all the actions are in italics. I kind of have it all set up that way. Serratus anterior is a good one to know, though. Right, serratus anterior, to reach or punch forward. Right, so if you extend your shoulder, meaning your arm is up the front of your body, that's not serratus anterior. But if you reach forward from that position or punch forward from that position, that's the job of serratus anterior, right? Which we saw under the arm is where it's located. Let's see here. Trapezius, large back, neck, and shoulder muscle, right? Shrugs shoulders, extends the head. You can say elevates shoulders, extends head. Large prime mover, so good one to know there. Levator scapula does exactly what it says. It elevates the scapula. And the rhomboid muscles adduct the scapula, meaning bring it towards the midline. If you remember where the rhomboid muscles are located from lab, well, then we can kind of work it out that if these rhomboid muscles contract, that's going to pull your scapula towards the midline, and that's called adduction. So some big prime movers to know some actions: the pectoralis major, shoulder flexion, and adduction. Basically, that's putting your arms together in front of your body. Where latissimus dorsi is shoulder extension and adduction. That's bringing your arms together behind your body. So there, when one's the prime mover, the other is the antagonist. Deltoid, always a good one to know. Shoulder abduction, that's basically raising your arm out towards the side. Not too worried about actions here because it's the rotator cuff group of muscles, but you should know the group of muscles that work on the shoulder to rotate it. So these are the four muscles that are part of the rotator cuff group: supraspinatus, infraspinatus, teres minor, and subscapularis. Now the big thing here is teres major is not part of that group, right? Teres major is its own muscle that I label it in lab. Don't worry about it too much here. But these are your four that you need to group under rotator cuff. Coracobrachialis does the same thing as pectoralis major, flexes and adducts the shoulder, which is arms together in front of your body. And when you see two things here, my lab students know this. An action on the arm is the same thing as the shoulder. Shoulder is the joint, but the arm moves as a result. So you'll see that slash there. You can use either one. Elbow goes with forearm, head goes with neck, hip goes with thigh, knee goes with lower leg, right? So when you see that slash, either of these parts of the body could be used, either the joint or the limb that's moving as a result. Right? And there, teres major, probably you just should know that it's not one of the rotator cuff muscles. All right, now these anterior arm muscles, a lot of brachii, right, in there, which means arm. So we have a biceps brachii, a brachialis, and a brachioradialis. All of them flex the elbow, which means bend the elbow. All right, we got elbow flexion, flexes elbow, same thing. But of these three, the biceps brachii has an extra motion, and that's supination, which means turn the palm upwards, right? Rotate your forearm to turn the palm upwards. So these are your elbow flexors, and the biceps brachii also supinates. This is the antagonist of those muscles that we just went over, those brachii muscles. Well, this is the triceps brachii. It's on the posterior side of the arm. Therefore, it's not elbow flexion. It performs elbow extension. All right, so if you're asked what's the antagonist of brachialis, for example, well, that would be the triceps brachii, because if one flexes the elbow, the antagonist extends it. All right, and these muscles aren't a big deal. They're just named after their actions. The pronator teres pronates, the supinator supinates, and those are motions that turn the palm upwards or downwards that we know from previous chapters. And when it comes to wrist and ankle, these are just kind of testing your medical terminology. Not too worried about the last word for lecture anyway. And if you see a muscle there and it's asking for its action, we should be able to work that out, that flexor carpi whatever flexes the wrist. Palmaris longus we can't do that with. Flexor carpi flexes the wrist, flexor digitorum flexes the fingers, flexor pollicis flexes the thumb. So we're good there. Same with these. Extend the wrist, extend the fingers, extend the wrist, extend the thumb, extend the index finger. Probably won't get that one because it's not on our lab list. And abducts the thumb, because pollex is our regional term for thumb, so pollicis has to do with the thumb. All right, some good groups here. So now we're at the hip or thigh. I like using the joint versus the body part there. So these are the muscles that flex the hip. So your main group of muscles that flexes the hip is your iliopsoas group. They perform hip flexion. And that's a group that's made up of iliacus and psoas major. And of course, we saw that in lab. These right here, I would kind of hold off on it, flexes the hip. If you just want to remember one muscle or one action for the quadricep femoris group of 4 muscles, it extends the knee or straightens the knee. So first, know the group of 4 muscles that belong to the quadriceps femoris group, rectus femoris, and the 3 vastus muscles, lateralis, medialis, intermedius. So those belong to the quadriceps femoris, which extend the knee, where the iliacus and psoas major belong to the iliopsoas group, which flex the hip. So we have 2 good groups of muscles, know the groups, and the action. Sartorius, if you remember cross legs, you're good, and that's a 3-in-1 deal, hip flexion, adduction, and lateral rotation. When you put all of these 3 together, it crosses one leg over the other. Hamstring muscles. Flexes, not all of them extend the hip, so I underlined this here for the one for you to remember. The hamstring muscles will first know the 3 hamstrings that belong to that group: biceps femoris, semitendinosus, semimembranosus. They flex the knee. So what's the I'll go back. What's the antagonist of the hamstring muscles? Well, since they flex the knee, the antagonist will extend the knee. So the quadriceps femoris group is the antagonist of the hamstring group. Gluteus maximus, or the butt muscle, the gluteus maximus, very forceful, but that action is hip extension. So its antagonist is that iliopsoas group. So iliopsoas group that you covered a second ago flexes the hip, while gluteus maximus extends the hip. These group of 5 muscles adduct the hip or thigh, you can use these interchangeably, and the ones we remember from lab are gracilis, adductor magnus, and adductor longus. There's a couple other adductors as well that were dropped from your lab list, but this is your group that adducts the hip or thigh on your leg towards the midline across your body. All right, so gluteus medius and minimus perform the same action, and which is abduction of the hip. So moving your thigh out towards the side. And you also have medial rotation for both of them as well. So gluteus medius, gluteus minimus, if you know one action, you know the other. Where piriformis does the other rotation. It laterally rotates the thigh. And then when we get to the lower leg, we see a tibialis anterior. So this is another good action. Dorsiflexion, that's raising your foot upwards, and inversion, turning the sole of your foot inwards. And it's like a 2-in-1 action for the tibia anterior. When you see muscles like this, it's named after its action. Again, extends the toes, extends the big toe for hallucis. The fibularis muscles, plantar flex, and perform eversion. So look at this: tibialis anterior, dorsiflex, inversion, fibularis muscles, plantar flex, eversion. So you can see how they're opposites of each other. So when one's the prime mover, the other group is the antagonist. So here are your 2 muscles that plantar flex, and that's at the ankle. That's like you're standing on your toes, and these are your calf muscles, gastrocnemius and soleus. And when we get to the end of the muscles here, again, just like in the forearm, we can easily pick out these actions. Flexes the fingers, flexes the big toe, and tibialis posterior kind of has its own little action. It's the prime mover of inversion, but I think that was our last one. Yeah, and I know I went through that quick. It's just most of you should know this, right, from lab already. I just wanted to highlight as we went through this the different groups. All right, so the big thing is just pay attention to which muscles belong to which group, and then the actions of specific muscles that I went over with you. Also, I was going to bring up one more point. I'm trying to think. Yeah, it's possible. I mean, you get questions randomly, so I never know what you're going to get. But you could be asked an antagonist question, right? Meaning, what's the antagonist of the brachialis muscle, right? So if brachialis flexes the elbow, then the answer would be whatever extends the elbow performs the opposite action, which would be, my example, the triceps brachii. One I see often is what's the opposite or what's the antagonist, let's say, of the iliopsoas group, like iliacus or psoas major. Well, that flexes the hip. So gluteus maximus would perform the opposite action, extends the hip. So that was the muscle group. I really want to get to the nervous system because there's a little more physiology here. So I'm going to pull up chapter 12. Oh, I need to share my screen again. I should check for questions, and I don't see any. So I will go ahead with chapter 12. Okay, so I'm just going to run through just the important concepts here with the nervous system. I'm going to start off real simple. You should know the difference between the central and the peripheral nervous system. And very simply, central is brain and spinal cord. Peripheral is spinal nerves and cranial nerves. If you ever see that word nerves, automatically it's the peripheral nervous system. The central nervous system does not have nerves. We call them tracts. They're the same thing, but structurally, but they're called tracts. So two types of cells in the nervous system: Neuroglia, or glial cells, which literally means glue cells, or nerve glue. There's a group of 6 cells that have specific jobs that we'll get to. We're going to name the 6 types of neuroglia and their function. So hold off on that. But the neurons are the excitable cells. I mean, they can send signals, electrical signals. Sometimes we call them nerve impulses. Sometimes we call them action potentials. But either way, those are the neurons. And that's what we're going to start with, the neurons. So neuron, as we know from lab, has a cell body, but it has all these processes sticking out of it. Well, the axon delivers that away from the cell body. Move that a bit, right? Where the dendrites, these processes send the signal into the cell body, right? So it's just one-way flow of information. And it says white matter and gray matter. Sometimes the axon, like you can see here, is wrapped in these Schwann cells, which are made of a fatty substance called myelin. If there is myelin on an axon, it is white matter just because that's its color. If there's no myelin, if you were to take all these Schwann cells off, it would look gray. So therefore, those are unmyelinated fibers. Okay. Here's a very good slide here as far as concepts go. Difference between a nerve and a tract, nucleus and a ganglion. Well, before I get into that, I should go to the next slide to explain what a nerve is. We already know what a neuron looks like. So there's the cell body. Of course, there'd be dendrites on there. There's the axons, right? The axons branch off at the end. We call that axon terminal, but that's fine. So this is a nerve. A nerve is taking all of these little axons, right? These little wires of thousands and thousands of neurons. Of course, there's only 5 here, but imagine 5,000 or 50,000 even. It forms this thick wire, You ever slice open a wire and there's all those tiny little wires inside of the wire? Well, the nerve is the whole thing, right? A nerve is made up of thousands of neurons, the axons of neurons, right? So if I go back here, a nerve is a bundle of axons, like we just saw, but only in the peripheral nervous system. In the central nervous system, we call them tracts. And distinction. Same thing, they're a bundle of axons, but it's a tract in the central nervous system and a nerve in the peripheral nervous system. Next, the cell bodies are located. By the way, if you're in my lab, we did this today. Where all the cell bodies are located, it's going to be thicker. So this nerve is going to bulge. You can see that right here, where all the cell bodies are located. And that bulge is called something depending on where we are. In the peripheral nervous system, it's a ganglion. And in the central nervous system, it's called a nucleus. The exact same thing, but just in a different part of the nervous system. So ganglion and nerve go with the PNS. Nucleus and tract go with the CNS. All right, nothing too testable here, but just to get into it, our nervous system will send information into the central nervous system, right? So these are nerves going towards our brain and spinal cord. That's sensory. Brain and spinal cord will take that information and process it so you understand what's going on. In this case, that's a glass of water. Right? It's processed somewhere in the brain. And then if it decides it wants to act on whatever it just sensed, that information coming in, well, then it'll synapse with a motor nerve, which will go to a muscle, perform an action, or a gland, which will secrete type of substance. Okay, so motor output is going to is something called an effector. So if I move back, whoops, sorry, I'm going the wrong way, apologize. If I go back, the effector here is the skeleton. Are either muscles or glands. Those are the only possibilities. So your motor output will either go to a muscle, skeletal, smooth, or cardiac, or a gland. And a gland will often secrete a hormone, which will make the body. There's also exocrine glands that we learned about in I think it was Unit 1. Yes, that secrete perspiration, right, for example. All right. So now we're in the peripheral nervous system. So it's not the brain spinal cord, right? Just the peripheral nervous system. Your nerves are divided into the sensory, like we said. We call that the afferent division. And the motor, we call that the efferent division. So then we can subdivide that sensory into somatic and visceral. So somatic, this is sensory information going towards the CNS, coming from things like your skin, so touch, pain, temperature, right? And from your skeletal muscles and joints. But your visceral sensory fibers are coming from your organs, like your stomach or intestines. So there's two types of sensory. There's also two types of motor or efferent fibers, somatic and autonomic. And we have a slide on each of these. All right, so your somatic nervous system, your effectors are skeletal muscle. And skeletal muscles are our only type of voluntary muscle. Therefore, right, that our only effector in the somatic motor division is the skeletal muscles, because we consciously control them, right? So this is the voluntary portion of the motor division of the PNS. Where right here, we have an autonomic division. Well, those are all the other effectors. Smooth muscle, we don't consciously control, neither do we cardiac muscle or glands. You don't have to think about it. You don't even realize they're contracting or secreting if we're talking about glands. So this would be the autonomic nervous system, or the ANS. Short forms are used on tests sometimes. So ANS is the autonomic division. So all effectors except skeletal muscles are used here. That's divided into sympathetic and parasympathetic, which is a whole chapter that we have on the ANS. That's chapter 15 in Unit 4. But sympathetic and parasympathetic just work opposite of each other. Sympathetic is your stress response, where parasympathetic is just your normal working nervous system. You're relaxed, digesting, sitting, reading division. Or sympathetic is, well, there's the S, but think of stress for S, exercise, embarrassment, emergency, excitement, all those E words for the sympathetic division. So, and I went through that kind of fast, but here is your entire summary of what we just went through, right? CNS, brain and spinal cord, that's done. PNS, that's your nerves, cranial nerves, spinal nerves, all the nerves in your body. Now they could be sensory, which means they deliver information to the CNS. You see the two blue arrows. If it's coming from your skin, like touch, pain, pressure, stuff you're aware of, that's somatic sensory. You can see that right there, somatic. Oh, that's motor, sorry, somatic. If it's coming from your organs, that's visceral sensory. All right, so there's the two types of sensory fibers, somatic, visceral. Usually we're not aware of visceral unless maybe you have a full, or you're hungry, maybe a full bladder, like things like that you can feel. Typically you're not aware of this sensory information from your organs going to your brain. Okay? Now the motor division is either somatic, meaning information to the skeletal muscles, right? You decide to do this, right? This is the only motor part or division that you have conscious control over, the somatic motor division, where motor autonomic, or the ANS, goes to other effectors like smooth muscle in your organs, cardiac muscle, and also glands that I don't think they're showing here. I don't see any. So careful here. So be able to organize that for yourself. I also forgot to mention that the ANS is sympathetic and parasympathetic, still going to cardiac muscle, smooth muscle, and glands, but one is for the stress situations and the other one's for regular situations. And we'll expand on that. We have a whole chapter on the ANS, sympathetic and parasympathetic, chapter 15. That's the last chapter we cover, I believe. Most common mistake here is not realizing the ANS is just motor, right? The motor division, ANS is only motor, but motor to what we don't consciously control: cardiac muscle, smooth muscle, and glands. All right, we've already seen this. There's our neuron, our nerve cell with dendrites. There's the axon. Neuron polarity just means information, electrical impulses, or can only travel in one direction, from the dendrites into the cell body and then out the axon, right? That's the only direction that information could move on a neuron. So dendrites send the signal into the cell body, where the axon delivers the impulse away from the cell body to the axon terminals, which will then synapse with another cell to relay that information to the next cell. Okay. Axon Hillock, this region right here, that's a very important region because that's where something called the action potential begins. Action potential begins, its electrical activity begins at the axon hillock and moves all the way to the axon terminals. Once it starts, it's going to go all the way to the end, like dominoes. Right? So an action potential either happens or it doesn't. If it does happen, that's where it starts, at the axon hillock. While I'm on the topic, that's not what this slide is for, but the signal coming in from the dendrites and into the cell body, that's not an action potential. It's not all or nothing. It's just a signal comes in. It's called a graded potential. That's a little later on in this chapter. We'll cover it. But the graded potential happens over the dendrites and the cell body. Now, that's not all or nothing, meaning as the signal comes in, it's going to die off. It's just going to weaken, and it might just die right about there. Right? But if that graded potential is strong enough to get to the axon hillock, right? As long as that axon hillock is stimulated, then you get the action potential. So the action potential is all or nothing. It travels long distances. where the graded potential on the dendrites and cell body, short distances, weakens as time goes on. It's a much different type of impulse. So action potential only on the axon, graded potential on the dendrites and cell body. All right. The nodes of Ranvier, that's just the spaces between the Schwann cell, and that's going to be important in a little bit. And the synaptic end bulb is just the tips of the axon terminals. They bulge at the end, no big deal. 3 types of neurons. I'll go to the picture for this so you have a visual. Yeah, these are most neurons in our body. They have a bunch of dendrites and an axon. So what we've been looking at. Few exceptions, some of our neurons are bipolar, meaning you have these dendrites, but there's only one fiber coming in and then one fiber coming out. That's the axon. And that's called a bipolar. We see that in a couple of our senses, in our eye and in our sense of smell, olfactory, in the linings. But that's it, only those 2 locations. And the unipolar, so basically you have dendrites on one side going into the cell body, but then it uses the same little branch as the axon to move out to the axon terminals. So there's only one process on the cell body versus 2 here versus many there. So this is a unipolar, and there's only one real special location which we see in lab, and that's the dorsal root ganglion. If you're in lab, you should know what that is. Maybe not yet. If you're not in my lab, you might not have covered that yet, but those neurons happen to be unipolar, but most neurons in your body are these ones that we've been looking at, the multipolar type. All right, so here are the neuroglia or glial cells. These are not nervous tissue, but other cells that have specific functions. They do not conduct action potentials. They don't have that ability. They're not excitable like the neurons are. But they do everything else. They protect the neurons, they wrap around them, they nourish them. So our job now is, 1, know which of these neuroglia cells belong to the CNS and which belong to the PNS, central versus peripheral nervous system. Well, the 2 S words, PNS, satellite and Schwann cells, where the rest go to the CNS. So that was the first job. Second job is match a function to each of these neuroglia. So first, astrocytes in the CNS. Astro means star. It kind of looks like a star. And see how they wrap around capillaries? They do this in your brain so that material can't leave the blood easily, because brain tissue is very sensitive. So it's very selective on what it allows out. We don't want toxins entering our brain tissue. So we have these astrocytes that it's not myelin. They just tightly wrap around capillaries to isolate the blood from the brain tissue. That's called the blood-brain barrier. So that's the main function of the astrocytes. Of course, that's just CNS. Microglial cells, you don't have to know what they look like, but they're phagocytes, meaning they eat up debris, right? If there's some microorganisms in the area, they can engulf it. Remember, phagocytosis means take material. So they're kind of the cleanup and take in any debris or microorganisms that might be in the area. So your keyword is phagocytize or phagocytosis for the microglial cells in the CNS. The ependymal cells, we find them in the ventricles of our brain. So they have cilia, which helps move the cerebrospinal fluid, right? And next chapter, which is Unit 4, will cover what cerebrospinal fluid is, but it's fluid that circulates around your brain and spinal cord, and these ependymal cells help that fluid move along. You may have covered ventricles and cerebrospinal fluid in lab already, or you may have not. But regardless, we get more into cerebrospinal fluid. That's what CSF is here next chapter. But for now, match the ependymal cells to being able to circulate that fluid around the central nervous system. Oligodendrocytes are the Schwann cells of the CNS, because the Schwann cells that make the myelin are in that wrap around the axons are in the PNS. The oligodendrocytes wrap myelin around the axons in the CNS. And you'll notice there's no nodes. Remember we had the nodes of Ranvier? In the central nervous system, one oligodendrocyte will cover multiple axons. So it just makes myelin and covers the axons in a different way, and you don't have the nodes that you do in the PNS. So they create the myelin sheath in the CNS, where Schwann cells make the myelin sheath in the PNS. And the last one is satellite cells. They regulate the chemical environment, so that's the only real thing we need to match there. So these two on this slide, these two neuroglia, are the two that are found in the peripheral nervous system. And they're just showing us Schwann cells in the peripheral nervous system. All right, so if a neuron or an axon has a myelin sheath, it will protect that axon a little better, of course, because it's this fatty substance around the axon, so it insulates the axon. This is electrical activity moving across it, so it's good to have that insulation, but here's the big one, it's faster. So as that action potential propagates or moves down the axon, it's much faster if there's myelin on it versus if there was no myelin on it. So the white matter, which means myelinated, transmit information at a faster velocity, right? Increased velocity versus unmyelinated fibers. So non-myelinated fibers condenses more slowly, right? But myelin in the speed of the action potential. They're just showing us a Schwann cell. There's the nucleus. It just keeps wrapping, kind of like a jelly donut. That's fine. All right, so here's the three types of neurons. I've touched on this already, but let's go through it. Sensory neurons. They transmit impulses toward the brain and spinal cord. So from all parts of the body to the CNS. Motor, just the opposite, carry impulses CNS to effectors. And we should know by now the effectors could be muscle or glands. Three types of muscle and glands. But then you have neurons entirely within, there it is, entirely within the central nervous system. Those are called interneurons, sometimes association, but we'll use interneurons. So they're mostly entirely, we could say 99% inside the central nervous system. They're the ones that are making the decision. If you remember that picture with the cup of water, right, sensory going in, then it says integration, right? These are the neurons that are deciding what to do. Should they act? Should they decide to act? For a neuron to carry out an action. So they get information from the sensory neurons, and if they decide to act on it, they will send synapse with a motor neuron, which will influence an effector.
Dr. Greco, can you?
Yes.
In the video, there wasn't anything about the glass of water, so can you fill that gap in for us?
Oh, okay. Yeah. No big deal here. That's what I showed you earlier.
Oh, I see. Thank you.
Yeah, that would be your sensory input, right? So that if we're using the terms that the slide that we were just on before, this would be your interneuron. It's in the central nervous system. So you have a sensory neuron or afferent neuron, afferent, going in, synapsing with an interneuron, which are in your brain and spinal cord. Now your interneuron will decide what to do. If it decides to act, it will synapse with a motor or efferent neuron, which goes to an effector, which is a muscle or gland. In this case, it's a muscle because obviously the interneuron decided it wanted that glass of water, so it synapsed with the motor neuron, which will go to a specific muscle. It would be a group of muscles, of course, but this is just a simplified illustration.
Okay, thank you.
Okay. So we know neurons are excitable. That's good. They can produce an action potential, right? And an action potential is always the same on a specific neuron, regardless of the stimulus. Like we said, an action potential either happens or it does not. We'll get to more of that in a second. we don't really need to talk about leakage channels. So I know a lot of, if you read through the textbook, don't worry about that too much. I'm going to go to the picture on the next slide to explain just the different types of channels. All right, so what we're looking at here is a plasma membrane. All right, you can see the phospholipid bilayer, which we learned in Unit 1. We know that in the phospholipid bilayer, sometimes we have these big proteins that are like channels. See how the channel opens up, right, which can allow substances to move into the cell, to move out of the cell. And we're talking about neurons here. So the neuron has a plasma membrane, right? So it can allow ions like sodium ions, potassium ions in or out. So this is just saying if a channel is chemically gated, like these here, when a chemical, there's a receptor, this protein is a receptor, meaning it receives something. So now a chemical has bound to that receptor, which causes the tunnel to open. So the gate opens in response to a chemical, right? So there's your little chemical, and those little chemicals are called neurotransmitters. When we covered muscle, that neurotransmitter was acetylcholine, right? That caused we called it the end plate potential in that chapter. But this is just saying that, yeah, some channels open up because a neurotransmitter is bound to it, right? And now allowing ions to flow in and out. Other channels are voltage gated, meaning the gate only opens if there's a charge or voltage rushing across it. So here's our cell, right, our neuron, positive on the outside, negative on the inside, and we'll kind of figure out why in a second. But look what happened here. Now it's negative on the outside, positive on the inside. That means a charge was rushing across this membrane to reverse it, right? The membrane voltage just changed. They flipped from here to here. So that voltage gated channel, the channel opens, see, sodium was not allowed in, right? But here it is allowed in because that voltage change causes the channel to open. So this is how ions flow in and out of the cell. And that's what changes the charge on the inside versus the outside of the cell, are these ions that are able to flow through when the channels are opened. And channels can open because a neurotransmitter is bound to the channel. We call that, well, the channel is sometimes called the receptor because it's receiving the neurotransmitter, or the gate can open because of the voltage that's rushed across the cell membrane. All right, and this is just saying when gated channels are open, the ions diffuse, from high concentration to low concentration. Yeah, and this is getting into resting membrane potential right now. So at rest, our neurons, our plasma membranes of the neurons, is the voltage or millivoltage is negative. It's actually negative 70. And it's because of the different ions in the intracellular fluid versus the extracellular fluid. I might stop here for a second and go to and draw something. Can you see my whiteboard?
Yes.
Okay, thanks. I could see the chat now. I think we're missing a video for some slides in this chapter. Oh, okay. All right. If you notice that some slides don't have a video, if you have the PowerPoint, you have a module that's just PowerPoints where you actually pull up the PowerPoint. If you let me know which slides you don't have a video of, I can look into that tonight and make sure I post that video. So just kind of let me know that, or if I remember after this, I'll check and see if there's a video missing. So. All right, so you could see the whiteboard here. All right, so we have a neuron. There's the cell body, the nucleus. Excuse my drawing, right? You got all the dendrites, right? One side you got the axon, right, coming out, right, and then all the axon terminals, right? So if I just take a little piece of axon right there and I enlarge it, slide 26 38. I'm going to write that down. 26 to 38. All right, so it looks like you're missing slide 26 to 38 video on that slide. So I will look into that and hopefully post it. The slides that talks about gates and membrane potential. Okay, yeah, I'll do that. I just enlarged this so you're looking inside the cell, right? This is a plasma membrane. So this is your intracellular fluid, right? There's your extracellular fluid, right? So you know what you're looking at. This is inside the axon. That's outside the axon. Well, you might remember, I'll make it a little bigger, the sodium-potassium pump. Potassium pump pumps potassium into the cell, and it pumps ions outside the cell. Right, so there's your plasma membrane. There's going to be gated channels, voltage gated channels coming in there, but I'm not drawing detail in the plasma membrane. So these are both positive ions. And the thing you might remember with the sodium potassium pump, because we did it, I think, in an earlier unit, is that 3 sodium ions move out for every 2 potassium ions that move in. So knowing this, we can see that there's more positives leaving, 3, than entering, 2. So every 3 that go out, 2 positives come in. So more positives are in the extracellular fluid, more positive charges, versus the intracellular fluid has more negative charges. As a result, the inside of the membrane on your axon, right? Actually, all of it on your inside your plasma membrane and your entire neuron is negative compared to the positive exterior. Excuse me, not great at drawing with a mouse, but actually I'm not great at drawing with pencil either. But, so this is what we get. So there's a charge difference. We call that voltage, right, across the membrane. And there's quite a few reasons for that, but one of the main reasons is because more positives are leaving than are entering, right? So when you're asked about a resting membrane potential, it happens to be -70. You're not going to have to repeat the exact number, but that was what we just saw, resting membrane potential in millivolts, MV. So when this neuron is not doing anything, when it's at rest, it's not firing impulses or anything, no action potential. When it's at rest, the resting membrane potential is negative. And they always use the inside as the measurement. It's negative because, well, one of the big reasons is because of that pump. All right, so at rest, we have this -70 voltage, millivoltage, on the inside of our membrane. Now, because of this voltage, this cell is excitable, so it's able to produce graded potentials and then action potentials. So good thing to know on this little drawing here is that if you're asked the difference between intracellular fluid and extracellular fluid, well, big thing is cellular fluid is more negative and contains more potassium and less sodium. There's still sodium in there, it's getting pumped out, but more potassium's moving in, and less sodium. So the extracellular fluid is, well, the plasma membrane on the outside is more and it contains more sodium ions and less potassium ions, just the opposite. All right, so if you know one, you know the other. It's because it's the opposite. I'll say it one more time. Intracellular fluid, or in the inside of the membrane, in the intracellular fluid is negatively charged and contains more potassium ions but less sodium ions. And it'd be opposite for the outside. Right, so these pumps are using ATP to move the ions across. What happens if I open a channel right there that allows the ions to move? Diffusion says they're going to move from high concentration to low concentration. Right, so whenever a channel opens that allows these ions to move through, that channel opens, sodium is going to rush in, right? Because you're going to have a high concentration outside, and diffusion says it moves from high to low concentration. Right? And if a potassium channel opens, potassium is going to rush out because there's so much of it inside that it just floods out. right? So that's kind of what happens during an action potential, and that changes charges here. No. Okay. Let me go back to the PowerPoint. Here we are. Right, so it's just saying the resting, oh, let me get my pointer. Resting membrane potential, yeah, about it is negative, negative 70, but you won't have to repeat that number, right? And it's generated by a few things, but we here they're just showing, yeah, the inside is negative, the outside is positive. I guess I could use this picture to draw on, and that it's a negative 70, and they always measure from the inside, and that's just up the axon. Remember, this is at rest. This is the resting membrane potential. Right, and this is saying the extracellular fluid, more sodium ions. Intracellular fluid has more potassium ions. And of course, we need to know that inside is negatively charged, outside is positively. And I know it says a bunch of stuff here, but the main reason why we get that positive and negative charge on either side is that sodium-potassium pump. 3 sodiums pumped out, but only 2 potassiums pumped in, and that creates a gradient. This is just the sodium-potassium pump. Don't worry about that too much there. All right, so next, there's 2 types of signals, and I already mentioned this. You have graded potentials and action potentials. You might recall that the graded potentials are only, I'm going to write this in. Right? It says it somewhere else, but since we're talking about only on dendrites and cell bodies, right? On neurons, they're only on the dendrites and cell bodies, where the action potentials only on the axon. Let's speak again for you. All right, so graded potentials, they're just work over short distances, and this is the electrical impulse that happens on dendrites and cell bodies. But when you get to the Axon Hillock, right now we're on, and that's when an action potential could be generated. So action potentials are long distance signals. They're all or nothing. They either happen or they don't. And they're only on axons, right? They start at the axon hillock and they end at the axon terminal. And I'm just talking about neurons here. We learned that the sarcolemma on muscle fibers can produce action potentials as well. So those are the only 2 membranes that action potential can happen on, right? On the neuron, only over the axon, and on our muscle fibers, only on their plasma membrane, which we call the sarcolemma. The word depolarization means we have a greater chance of producing a nerve impulse, right? Whether it's a graded potential or action potential. Here's negative 70. By the way, you get a nerve impulse when the negative becomes positive, right? When we cross the 0, now that's a nerve impulse, so these charges flip. So right here, look what happened to the membrane right here. It was at rest, negative 70, but all of a sudden, oh, we got closer to 0, right? That's called depolarization. Now it's more likely to produce an action potential than at rest because, well, we're less negative now, right? We climb to what, maybe negative 60-ish, right? So because we got closer to the positive side, that's called depolarization, right? It's moving toward 0, right? Getting less negative. So something like this would be depolarization, heading towards the 0. If it's hyperpolarized, that's the opposite. So hyperpolarization reduces the probability of producing a nerve impulse because look, here's our negative 70 at rest, resting membrane potential, but now we got even more negative, right? So if it's more negative, we're further from the positive or further from 0. So it's becoming more negative, therefore it's more difficult to produce an action potential. So that's just measuring on a dendrite right there. So here's everything on graded potentials. They can either depolarize, which is make an action potential more likely, or they can hyperpolarize, make an action potential less likely. But with graded potentials, once that current flows, it's going to quickly die. So they only work over a short distance, and this only happens, like I wrote in earlier, on dendrites and on the cell body of a neuron. Right? So they're just showing there's the signal coming in, right? It's depolarized, but as we move away, it just dies off very, very quickly. So if I go to a nerve here, so if you have a graded potential coming in, remember it has to get to the axon hillock, and if it does, you get the action potential. And once the action potential happens, it doesn't stop. It's going to go all the way to the axon terminal. But one little graded potential is probably going to die off before it gets to the axon hillock. So what's tiny little signal is not going to do anything. We need many signals coming in, right, that add on to each other, and we'll get to that. It's called summation, but we'll get to that in a minute here. So we're starting to put it all together. So an action potential now, not a graded potential, only occurs, well, specifically on the sarcolemma of muscle cells and on axons of neurons. They don't decay like graded potentials do. They either happen or they don't. So we're going to go through the steps of an action potential. I'm going to go all the way to this picture here because it's larger than the ones on the slides. All right. So here's step one, which is not really a step. It's the resting membrane potential. We can see we're -70. So what happens at the axon hillock? If threshold is met at the axon hillock, if the graded potential reaches the axon hillock, it'll start to depolarize. And once you get to about -55 or so, again, I won't ask you numbers, that's called, there's a threshold. Well, once it starts rising, once it hits the threshold, it's going to happen, right? It's going to go all the way up to the positives. You see +30 there. So this is called depolarization. And what's happening is sodium is rushing into the cell. Remember, there's more sodium ions outside the cell. So depolarization, the sodium channels open, much outside, it's going to flow into the cell. And all those positives rushing into the cell, all those sodium ions, they're positive, makes it go from -70 all the way up to -30. And that's called depolarization. So we just switched the charge. The inside was negative, now it's positive because of all those sodium ions rushing in, because the gate opened, right? The sodium gate. Split second later, potassium gates open. They're a little bit slow to respond. So now the potassium gates open. Remember, potassium ions are inside the cell. So they start rushing out of the cell. And since positives are now leaving the cell, we go all the way back to negative again. So here's the resting membrane potential. Sodium channels open, you get depolarization. Potassium channels open, you get repolarization. One more little step, that repolarization, meaning back to rest. There, repolarization, back to rest. It overshoots, right? That's the resting membrane potential right here. It actually gets more negative for a small period of time. And that's called hyperpolarization, because remember what that means? Something's hyperpolarized, it's more negative than at rest, which means it's less likely that an action potential will happen because now we're further from zero. So those are the 4 steps: resting, depolarization, repolarization, then hyperpolarization. And you should also know that the sodium channels open in depolarization, so sodium rushes in. Potassium channels open in repolarization, so potassium rushes out. Okay. This is just saying an action potential either happens completely or not at all. It's all or nothing. All right, so if an action potential is always the same, it either happens or it doesn't, one way for our brain to know that the stimulus is more intense is by just firing more action potentials. So that's a real important point. I'll show you a picture of this, right? Let's say something landed on our skin here, but it's too tiny, we didn't even feel it, right? It didn't even reach threshold. So your brain is not receiving any action potentials. But now let's say a fly lands on your skin. Well, now that's strong enough to reach threshold, right? So your brain is going to get a certain number of action potentials, letting you know the intensity of, my example, whatever landed on your skin, right? Now, something much heavier just landed on your skin. Well, how does your brain know that it's a more intense signal or stimulus? Well, it fires off more action potentials one after another, right? See, all the action potentials are the same, right? But more of them are occurring with a higher intensity of stimulus. So the point is, well, sorry, wrong slide. How does your brain know if the stimulus is stronger? Well, there's a higher frequency of impulses, and that codes for a stronger stimulus. All right, a couple more points to make in this chapter. I think I'm over an hour. I apologize, but probably just need 10 more minutes here. There's a couple periods. We already talked about the latent period in last chapter, a couple chapters back. That's the excitation contraction coupling. That was with muscle. But there's two periods in this chapter, two refractory periods, absolute and relative. Here's the difference. During this time, during depolarization and repolarization, what you see in green here, another action potential cannot happen, right? That nerve is firing off an action potential. That means it's impossible to have another action potential right here. That's the absolute refractory period, right? It's in no way is it possible for another action potential to happen during this point in time. I just used to think of it as there's absolutely no way, right? That's the absolute refractory period. This area in dark blue, where you have the hyperpolarization, is the relative refractory period. That means that you can have another action potential right here if you want to, but it's just going to be more difficult to do so because we're more negative during hyperpolarization than the resting membrane potential, right? So absolute refractory, no action potential possible. Relative refractory, it is possible. You just need a really strong stimulus for it to happen, right? Only an exceptionally strong stimulus can stimulate one. A couple quick things. We already mentioned that a myelinated axon will conduct an action potential faster than an unmyelinated one. Here's why, because it uses, here's a good word, saltatory conduction. Saltatory means to jump or skip, right? So if you have myelin, right, that action potential just skips from node of Ranvier to node of Ranvier, right? It just skips from node to node to node. And that's a much faster action potential that this, compared to this axon, which has no myelin, well, that channel has to open, and that channel has to open, and that channel, right? You have a lot of channels that have to open one after another. That takes time, right? But in a myelinated fiber, the channels are only located in the gaps, the nodes of Ranvier, right? So we have the stimulus, well, then it just skips from node to node, which makes a much faster action potential, not contraction. And it's called saltatory, right? Because of the skip of the action potential between nodes. All right, I'm going to go to the last part here. Yeah. So we're talking about graded potentials now. So remember, you need a graded potential that's strong enough on the dendrites and cell body to reach the axon hillock before you can get an action potential. So I'm going to go here. So these are two types of, doesn't say it here, but should, right? Graded potentials. They can either be excitatory or inhibitory. So we call them EPSPs or IPSPs, excitatory or inhibitory postsynaptic potentials. So with excitatory or EPSPs, well, more sodium coming in than potassium going, that's fine. But what that's saying is if it's a graded potential that's moving close, well, it's excitatory, right? Because we're getting closer to threshold. If it's inhibitory, well, then we get hyperpolarization, right? So you want to match hyperpolarization to inhibitory because it's harder to produce an action potential if the graded potential is making the membrane more negative than the resting membrane potential. So the point is that one little graded potential cannot create an action potential, right? It'll never bring the Axon Hillock to threshold. So that means we have to add graded potentials together. We need more than one of them adding together, and that's what's called summation, right? A sum is when you add. So summation is we need to add a whole bunch of excitatory postsynaptic potentials together in order to get an action potential. So there's two ways you can do that. And this is the last main thing that you need to know for the exam. Temporal summation and spatial summation. So here's no summation, meaning here's a little graded potential. It's excitatory, that's the E, right? So what happens? Oh, I got my little depolarization, and then it fades. Send another signal in. Oh, there's our depolarization, and then it fades. So it was never enough to reach threshold. So we can never get the action potential just with nothing adding on to it. So there's no summation. But temporal means time, right? So temporal summation. What if we send in two signals close together in a very small amount of time? Again, this is so quick that we can't even, it's instantaneous to us. But so there's one graded potential. It's excitatory, so it's depolarizing. But before it can rest, right, during that that relative refractory period, right about there, we send another signal in. Well, one piggybacks on the other. So as soon as you hit that negative 55 there, again, you don't need the numbers, but you're going to have that action potential, all or nothing. So we just added two graded potentials together. We summated them, right, summation. But this is temporal summation because two fired off in a small period of time, right, temporal. Another type of summation is spatial summation. That means, well, let's send a graded potential in here at this dendrite and another one in here at the dendrite. So it's not temporal, meaning this isn't firing twice, it's just firing once, right? And this one is just sending an impulse once. So you have two excitatory graded potentials at the same time, but apart from each other, and that's the spatial summation. So one from one side, another from another side, they add together to reach threshold, and then you get your action potential, right? It starts at the Axon Hillock and rushes down. So this is just very basic. Two would not even do it. You would need like tens or dozens or hundreds, right? Your neurons stimulated all the time. Sometimes with inhibitory, right? So here's an excitatory, here's an inhibitory. Summate those two, spatial summation. Well, they sort of cancel each other out, so you never even get close to threshold. So neurons are always receiving these EPSPs and IPSPs, but it's only when a whole bunch of EPSPs come in, spatial summation, temporal summation, that they can all add up to reach threshold at the Axon Hillock, right, to produce the action potential. I'm not worried about this last stuff, neurotransmitters. You should check the questions. I know we've been through a lot there. All right. Oh, and there was none. Okay. Yeah, that's just a good recap. 12 is kind of a beast, right? Of a chapter, but luckily there's 3 other chapters in Unit 3 test. I don't see any questions. I will hold out for a second. Oh, something's here. How do we get the recording after this is over? Let me show you. Someone just asked, how do we find this recording? I'll share my screen. All right, I'm just going to go at Blackboard. Sorry. Oh, that's banner web. Whoops. Let me go into, wow, I just hit banner web again. Wow. Blackboard. Okay, there's our class. So to get to the recordings, go to Blackboard Collaborate Ultra, just like you did. Oh, you can see my screen, I'm assuming. Yep, it says you're sharing your screen. When you get into Blackboard Collaborate Ultra, there's these 3 little lines right here, the menu. This is where you went into the course room, but just click on those. And then here's the recordings. Well, I'm not sure why it says nothing in the past 30 days. Oh, you know what? I went into the wrong class. That's why. Class. Then we'll go into Blackboard Collaborate. There's the menu, recordings. There they are. So those are the other two review sessions right there that you can play. And then you'll find this one probably within a couple hours. It'll appear down there. So. Okay. Okay. Is there a chapter that has more of a focus? Now they're all equal, even though some chapters seem more intense than others. They're more or less about the same amount of questions for each chapter. And it's like that throughout the course. So Unit 4 will be the same. They're about to be the same amount of questions for each chapter. All right. All right, well, I'll just hang out for a few minutes until everyone hangs up, logs off, and then I'll go in and slide 26 to 38. And if there's a missing video, which obviously there is, I'll try to find it and post it. Or if it's slides or I thought maybe didn't need a video, I doubt that. I'll let you know, but as soon as we get off here, I will look into that. All right. Is there a final quiz for? There's no quiz unit exam for, but that's probably what you meant. So this is unit exam 3 this weekend. We have unit exam 4 coming up pretty quick. Then there's a final on everything. All right, so the final exam is on chapter 1 through 15, which is all 4 unit exams sort of put together. And it's 100 questions instead of 50. And it ends up being like 5 or 6 questions a chapter on average, I think, something like that. But I'll send an announcement about the final. It's multiple choice as well. So. All right, you're welcome. Yeah, and I'm just waiting to see if there's no questions. Oh, will there be a study? The PowerPoints videos are the study guide. I don't know if you ever read the chapter. The chapters have a lot more information than I have in my PowerPoints and videos. That's as much as I can possibly cut material down. I kind of walk the line on not enough versus enough. I just try to include only what's has to be there. So yeah, the study guides are my PowerPoints and videos. If I cut anything else out, you would be missing stuff that was on the finals, or could be on the final, I should say. All right. Good. So we hit a lull in the questions. So I guess I'll just hang out or log out when everyone else logs out. That's the way I know there's no more questions. And good luck this weekend.